Somewhere back near the very beginning, there was light. And whether you believe in creation or evolution, or some combination of the two, life appeared on the scene. Primitive Neanderthals abandoned the cave and set out on the road to civilization. And today, as civilization continues to grow, the demand for energy is placing an unbearable strain on traditional fuel reserves. The need to harness the unlimited energy of the sun is increasing with great urgency. But while our efforts to capture the sun's energy economically have so far proved inefficient, plants have been successfully harnessing sunlight for eons. In sunlight, green plants combine carbon dioxide and water to form organic molecules. These molecules, called carbohydrates, provide material for plant growth. Green plants can be thought of as complex, self-sustaining machines, converting solar energy into chemical energy. Man-made solar collectors can't hold a candle to them. It was a candle, in fact, that helped English chemist and clergyman Joseph Priestley to discover in 1771 some of the remarkable things that plants can do. From Priestley's diary, I flatter myself that I have accidentally hit upon a method of restoring air, which has been injured by the burning of candles, and that I have discovered at least one of the restoratives which nature employs for this purpose. It was vegetation. Priestley found that candles go out when put in a closed jar, but burn well in air that plants had restored. He then replaced the candle and obtained, unfortunately for the mouse, the same result. Priestley's experiments showed for the first time that plants release oxygen, though all he needed was that the plants replace something the mouse and candle had used up. But he wasn't aware that without light, plants couldn't restore the air. His findings sparked interest in the scientific community, leading to further discoveries. Although water was known to be essential for plant growth, it was not until 1779 that the Dutch physician Jan Ingenhousz demonstrated that sunlight is required before plants can give off oxygen. Ingenhousz also showed that only the green parts of plants are involved in the phenomenon which became known as photosynthesis. A few years later, the Swiss scientist Jean Senevier furthered the understanding of photosynthesis. He discovered that the whole process depended on a particular gas, carbon dioxide. So, by the early 19th century, scientists were ready to close this chapter on the subject. They had assumed that they had identified the basic reactants and products of photosynthesis. However, it took another century to discover that not only did plants require water, they produced it as well. Green pigment, called chlorophyll, absorbs light energy and converts it to chemical bond energy, ultimately resulting in the production of organic matter or carbohydrates. Substituting symbols for words, the equation now looks like this. Perhaps the most significant thing that happens during photosynthesis is that the carbon dioxide breaks down, and the carbon is fixed as, or becomes part of, organic matter. The other lines show the fate of the rest of the atoms involved. Brackets around CH2O show that it's only a grouping. It takes 6 of these groupings to make one molecule of the 6-carbon sugar glucose. so. Multiplying the equation by 6, the glucose molecule, C6H12O6, is now complete. While water appears on both sides of the equation, the water on the right-hand side is actually new water. This was proven by feeding plants water containing the radioactive isotope oxygen-18. Only half of the hydrogen from the old water goes into forming the new water. The rest forms part of the glucose. The oxygen from the old water is only used to form oxygen gas, which is released during photosynthesis. The oxygen in the new water comes from the carbon dioxide. But the process is far more complex than this summary equation shows. A lot happens from the time sunlight first shines on a plant to the production of enough carbohydrate molecules to grow a leaf. Current evidence indicates that photosynthesis occurs in two distinct stages. The first stage is dependent on light and is called the light reaction. It is the light reaction which uses the old water and gives off oxygen as a waste product into the surrounding environment. The second stage, the dark reaction, doesn't actually require darkness. It simply doesn't need light. What the dark reaction does require is carbon dioxide, as well as the products of the light reaction, except for oxygen. The dark reaction uses these substances to produce new water and supply the energy needed to fix or incorporate carbon from the original carbon dioxide into the CH2O groupings, which then, in clusters of 6, form glucose, one of the simplest carbohydrates. These carbohydrates constitute organic matter and make up most of the plant's leaves, stems, fruit, and flowers. And so the plant grows and matures. Plants then can be thought of not only as the machinery, but the product as well. An important part of the product and the basic part of the machinery of photosynthesis is the leaf. It is the chlorophyll in the leaf that absorbs light used for photosynthesis and gives plants their green color. Chloro from the Greek word for green, and phyle meaning leaf. In most plants, chlorophyll is found in a layer of cells called palisade cells, located just under the leaf's upper surface. Palisade means fortification, and what the palisade cells enclose are special membrane-bound bodies called chloroplasts. In a palisade cell from a tobacco leaf, shown here in cross-section, the chloroplasts line the inner membrane of the cell. Inside each chloroplast is an elaborate system of long, thin membranes called lamellae. And it's here at last where the chlorophyll is found. In 1771, English chemist Joseph Priestley's experiments with plants sparked interest in the scientific community in what would later be known as photosynthesis. Most green plant photosynthesis takes place with the help of chlorophyll, just beneath the leaf's surface in special bodies called chloroplasts. The green pigment chlorophyll lies within the chloroplasts in long, thin membranes called lamellae. And spaced out along these membranes are disc-like bodies called thylakoids. Thylakoids tend to occur one on top of the other in groups called grana, which look a lot like stacks of coins. In fact, grana in Latin means stacks of coins. The first major stage of photosynthesis, the light reaction, occurs here in the thylakoids, and everything required for the light reaction is found in the thylakoid membranes. This then is the setting. All we need now is light. Light falling on a leaf is either absorbed, transmitted, or reflected. When we look at an object, the color we perceive is the portion of the light spectrum that the object reflects and transmits. Because a leaf appears green, it must be reflecting and transmitting almost all of the green portion of the spectrum. This suggests, therefore, that wavelengths other than green are being absorbed by the leaf. Indeed, the red and violet wavelengths are absorbed almost entirely. It is chiefly the pigment chlorophyll that reflects green wavelengths and absorbs red and violet wavelengths. In fact, two different forms of chlorophyll play an important role in photosynthesis, with chlorophyll A being the more important of the two. The only structural difference between them is that this CH3 group in chlorophyll A is replaced by COH in chlorophyll B. These two green chlorophyll pigments are not the only pigments found in plants. Colorful gardens and fall landscapes offer proof of that. Other pigments called carotenoids give autumn leaves and most fruits and vegetables their color. The carotenoid beta-carotene, for example, gives carrots their bright orange hue. The amount of green chlorophyll in plants and trees depends mostly on light and temperature. As autumn progresses and the days become shorter and cooler, the chlorophyll pigments are the first to decompose. Red and yellow pigments, like the carotenoids, remain behind, covering the landscape with a blanket of vibrant color, until they too become casualties of the approaching winter. With the return of spring and hot, sunny days, the growth cycle begins again. While chlorophyll A absorbs the red and violet wavelengths almost entirely, other pigments, which include chlorophyll B and the carotenoids, absorb other wavelengths. These pigments then pass the energy absorbed on to chlorophyll A, which uses that energy to drive the light reaction of photosynthesis. So, besides giving us fall colors, the other pigments provide an energy boost to chlorophyll A, and in so doing, improve the efficiency of photosynthesis. Because of the way they trap and redirect the energy of incoming light, these pigments are called antenna pigments. An antenna on a rooftop intercepts television signals and passes their energy onto your set, while antenna pigments intercept light waves and pass their energy on to chlorophyll A. While you receive a better picture on your television set, chlorophyll A receives more of the sun's light energy to carry out photosynthesis. Even though chlorophyll B has nearly the same molecular structure as chlorophyll A, it actually functions as an antenna pigment. Despite the differences in function of chlorophyll A and the antenna pigments, they all share an important feature: alternating single and double bonds. These alternating bonds can be easily seen here with chlorophyll, and here with the beta-carotene molecule. The double bonds represent electrons less tightly bound to the molecule. And because these are weak bonds, it takes less energy to jolt the electrons out of their orbits. In photosynthesis, that jolt is provided by light energy. which exhibits properties of both waves and particles, consists of discrete bundles of energy called photons. Since each pigment molecule has very specific energy requirements, it only absorbs those wavelengths of light, or those photons, which supply the necessary punch. As we have seen, the antenna pigments, the carotenoids and chlorophyll B, absorb photons with energies other than those acceptable to chlorophyll A. The antenna pigments then pass the energy of the photons on to chlorophyll A in bucket brigade fashion. To do this effectively, the pigments are grouped together, so scientists believe, to form reaction centers, called photosystems. Since a photosystem contains many different pigments, and each pigment has a preferred wavelength, the combined effect is an overall average wavelength that the photosystem as a whole absorbs. This average wavelength is determined by the number of each kind of pigment present in the photosystem. As it happens, only two kinds of photosystems are formed, and each has a different average wavelength. For one, the average wavelength happens to be 680 nanometers, so it's called photosystem 680, or P680 for short. For the other photosystem, the average wavelength is 700 nanometers, so it's called photosystem 700, or P700. Both photosystems are needed to complete the light reaction. They occur in pairs within the thylakoid membranes, one P700 for each P680, forming what are called photosynthetic units. So, the photosynthetic unit is thought of as the actual physical unit that carries out the light reaction.
Great job, men. See you in the morning. Good night, Phil.
Good night, Phil.
Scattered throughout the thylakoid membranes, millions of photosynthetic units capture the energy of the sun in the light reaction process. Each photosynthetic unit consists of two photosystems, called P680 and P700. Green pigment chlorophyll A molecules in both P680 and P700 absorb photon energy. Chlorophyll A absorbs a photon directly if the photon has just the right amount of energy. If the photon doesn't have the exact energy required by chlorophyll A, then it is the responsibility of the antenna pigments around the chlorophyll A to absorb the energy and pass it on. As photons continuously bombard pigment molecules, it is the double bonds of the molecules that take the brunt of the bombardment. A photon strikes one of the electrons in a double bond, jolting the electron from its orbit. In order to continue absorbing photons, the molecule which has become electron deficient must constantly replace the lost electrons. These replacement electrons come from water, readily available within the thylakoid. In a process called photolysis, water molecules split, yielding two electrons, two hydrogen ions, or protons, and one atom of oxygen, which quickly bonds with a second oxygen atom to form one molecule of oxygen gas. Most of the oxygen is released into the atmosphere. The protons drift into the interior of the thylakoid, and the electrons are free to replace lost electrons. as the photon absorption process continues, those electrons lost by P680 are transported by special carrier molecules to the electron-deficient P700. The electrons lost by P700 are also passed on by carrier molecules to end up ultimately outside the thylakoid in a region called the stroma. This process of moving electrons from water to P680 to P700 and into the stroma is called the electron transport system. The electron transport system consists of an elaborate chain-like series of oxidation-reduction reactions. For our purposes, we'll think of the oxidation-reduction reactions in their simplest form. Oxidation means losing electrons, while reduction, the opposite process, means gaining electrons. Both oxidation and reduction must occur in order for the electron transport system to function. Each time one of the electron carrier molecules loses an electron, it is said to be oxidized. And the electron carrier that gains the electron is reduced. While the main purpose of the electron transport system is to move electrons through the photosynthetic unit, some of the carrier molecules perform an additional function. That is, they transport both electrons and protons in the form of hydrogen atoms. Because of this extra function, they are called hydrogen carriers. One of these special hydrogen carriers, plastoquinone, or PQ, located between photosystem 680 and photosystem 700, acts as a proton pump between the stroma and the thylakoid interior. The protons lie outside the thylakoid in the stroma, and the role of PQ, as well as transporting electrons, is to transfer protons from the stroma down to the thylakoid interior. However, PQ can't carry naked protons. It can only carry hydrogen atoms. So it overcomes this dilemma by picking up 2 electrons from the transport system, then 2 protons from the stroma, and combines them to form 2 hydrogen atoms. PQ, having acquired the 2 hydrogen atoms, has gained electrons and has therefore been reduced. The reduced PQ is called PQH2. The H2 signifies that it is indeed carrying 2 hydrogen atoms. Now the unique ability of plastoquinone comes into play. It physically moves or diffuses down through the thylakoid membrane to its inner boundary. Where it releases the 2 protons into the thylakoid interior and the 2 electrons back into the transport system and on their way to electron-deficient photosystem 700. Freed of its burden, the plastoquinone returns to start the process all over again. Beyond photosystem 700 are other electron carriers in the transport system. Energy-rich electrons from P700 move from one carrier to the next, until they arrive at the small carrier molecule, FAD, at the end of the electron transport system. Once FAD acquires 2 electrons, a molecule of NADP moves into position to pick them up. But NADP is a hydrogen carrier. So at the instant the electrons are passed from FAD to NADP, 2 protons from the stroma combine with the electrons to form 2 complete hydrogen atoms. Having gained electrons by acquiring 2 hydrogen atoms, the NADP has been reduced and is now called NADPH2. The reduced NADPH2 moves back up into the stroma. So, hydrogen carriers perform two separate tasks: the transport of electrons and protons. The protons released by the plastic quinone mingle with the protons liberated during the photolysis of water, gradually increasing the concentration of protons in the thylakoid interior. These protons play a critically important role in photosynthesis. As the proton buildup continues, pressure is relieved with the removal of surplus protons through special ion channels in the membrane. At the outer end of each channel is a large complex of enzymes called a CF1 particle. As the protons pass into the CF1 particle, it taps some of their energy to convert adenosine diphosphate, ADP, to adenosine triphosphate, or ATP. The whole of the light reaction has led to this, the production of ATP. ATP is so important that biologists refer to it as the power supply of life.
Join me. Come over to the dark side of photosynthesis.
Buried within the thylakoid membrane, millions of photosynthetic units carry out the first stage of photosynthesis by converting light energy to chemical energy. This first stage, known as the light reaction, releases oxygen into the atmosphere and produces NADPH2 and ATP molecules for the second stage, known as the dark reaction. The dark reaction takes place in the stroma and produces the final products of photosynthesis: glucose, cellulose, protein, and starch. It's called the dark reaction because none of the energy required for any of its individual reactions comes from the direct absorption of photons. Instead, its requirements are met by energy-rich products of the reaction. Since the light reaction requires light, and since the dark reaction depends directly on the light reaction for its energy needs, both processes can only occur during daylight hours. Beginning in the 1940s, American biochemist Melvin Calvin spent more than 20 years tracing the path of carbon in photosynthesis. He discovered that the dark reaction is actually a whole series of reactions responsible for fixing carbon from carbon dioxide into organic molecules. This series of reactions eventually became known as the Calvin cycle. Calvin experimented with chlorella, a single-celled green algae, because he could control its rate of photosynthesis in the laboratory. His experiments involved feeding the algae carbon dioxide containing radioactive carbon-14, then exposing the algae to light. To his amazement, he discovered radioactive carbon in the 3-carbon compound phosphoglyceric acid, or PGA, after only 5 seconds of illumination. Let's step through the individual reactions of the Calvin cycle, starting with one molecule of the 5-carbon compound ribulose phosphate, or RUP, which is present throughout the stroma. RUP encounters an energetic molecule of ATP, fresh from the light reaction. In reaction 1, the ATP donates its 3rd phosphate group to the RUP. The ATP minus 1 phosphate group becomes the slightly less energetic ADP, which then leaves the scene, returning to the light reaction for more energy. RUP has gained a 2nd phosphate group and is now ribulose diphosphate, or RUDP. The di in the diphosphate means two phosphate groups. Carbon dioxide, which has diffused into the plant, now enters the Calvin cycle. In reaction 2, one molecule of carbon dioxide combines with the RUDP, forming an unstable 6-carbon compound. In reaction 3, this unstable compound quickly cleaves into 2 molecules of phosphoglyceric acid, PGA, each with 3 carbon atoms. In reaction 4, ATP molecules once again enter the cycle. Each molecule gives up its 3rd phosphate group to a molecule of PGA. And each molecule of PGA, having gained the phosphate, becomes the compound DPGA, or diphosphoglyceric acid. In reaction 5, NADPH2, the other ingredient provided by the light reaction enters the scene. NADPH2 injects each molecule of DPGA with energy. Both DPGA molecules lose 1 phosphate group to become new 3-carbon compounds called phosphoglyceraldehyde, or PGAL for short. Only one main stumbling block prevents the 2 PGAL molecules from combining to form glucose. If we use the analogy of a key in a lock, the 2 PGALs represent 2 keys. One of them must be changed. And this is exactly what happens. With the assistance of enzymes, one of the PGAL molecules transforms into an unstable molecule called DHAP. Now the key fits the lock. The unstable DHAP quickly combines with the other PGAL molecule to form fructose diphosphate. A few more enzymes. The loss of 1 phosphate group, and glucose phosphate is produced. Glucose phosphate can be dephosphorylated, that is, made to give up its remaining phosphate group, to become, finally, glucose, the simplest sugar. If glucose is not immediately required in the plant, glucose phosphate is instead synthesized into starch for long-term energy storage. Let's briefly go back to the Calvin cycle. Reaction 5 produced molecules of PGAL. Surprisingly, only a few PGAL molecules go on to form glucose or starch. More than 80% are used to produce new ribulose phosphate, which finally closes an imaginary loop and completes the Calvin cycle. By looking at the Calvin cycle in an expanded diagrammatic form, its cyclic nature becomes more evident. ATP donates a phosphate group, converting ribulose phosphate to ribulose diphosphate. The addition of carbon dioxide forms an unstable intermediate compound, which quickly splits into 2 molecules of phosphoglyceric acid. Then, more ATP reacts with the 2 PGA molecules, giving them each an additional phosphate group and producing diphosphoglyceric acid. NADPH2 molecules from the light reaction infuse new energy and transform both DPGA molecules to phosphoglyceraldehyde. one of the two PGAL molecules is converted by enzymes to DHAP. It then combines with the remaining PGAL to form glucose phosphate. Finally, simple glucose is produced, and the unused PGAL molecules are reformed into RUP, bringing us back where we began. The Calvin cycle, named after biochemist Melvin Calvin, uses carbon from carbon dioxide and energy from the light reaction to produce materials for plant growth. By exposing green algae to radioactive carbon dioxide, Calvin first detected radioactivity in a 3-carbon compound called phosphoglyceric acid. He also noticed that many kinds of plants immediately fix carbon from atmospheric carbon dioxide into 3-carbon compounds. Calvin called such plants carbon-3 plants, or C3 plants. On paper, the Calvin cycle appears to be about 38% efficient. This means that, theoretically, 38% of the energy the Calvin cycle receives ends up in the chemical bonds of carbohydrates, with the rest of the energy lost as heat. But in reality, the Calvin cycle performs far less efficiently than expected. In fact, C3 plants can usually convert less than 1% of the light energy they absorb into carbohydrates. To understand why this happens, we need to take a closer look at carbon dioxide. One problem is the relative scarcity of carbon dioxide in the atmosphere, only 0.03%, as compared to oxygen at 21%. Carbon dioxide must compete with the more plentiful oxygen for the attention of ribulose diphosphate. Although RUDP is supposed to combine with the carbon dioxide to produce the unstable 6-carbon intermediate, it often joins with oxygen instead. The oxidized RUDP then goes through a series of reactions which eliminates it from the Calvin cycle. So, considering all the obstacles, it's no wonder that the ability of carbon-3 plants to fix carbon into carbohydrates doesn't measure up to expected efficiency. Around the time Calvin wound up his work, evidence was mounting to suggest that sometimes carbon doesn't first make its way into 3-carbon compounds. In plants generally found in tropical and desert climates, the first organic molecule in which carbon is fixed is a 4-carbon compound. Consequently, these are known as C4 plants. But why should C4 plants thrive in these environments? Let's begin to answer the question by comparing the two types of plants. In both C3 and C4 plants, the interior of the leaf is called the mesophyll. In C3 plants, the mesophyll cells, where photosynthesis occurs, are arranged in 2 layers. At the top is the palisade layer, and below it is the spongy layer, named for its sponge-like appearance. Embedded in the spongy layer are bundle sheath cells, which surround the network of veins spread throughout the leaf. In C4 plants, the mesophyll cells are tightly clustered in large rings around the bundle sheath cells. These bundle sheath cells are larger and contain their own chloroplasts. However, the grana inside are few and poorly developed, and are virtually unable to carry out the light reaction. So, it is left to the chloroplasts in the mesophyll cells, with their well-developed grana, to produce the bulk of ATP and NADPH2 for the dark reaction. And to make life even more difficult, C4 dark reactions require the involvement of both mesophyll and bundle sheath cells. Because they're so close, materials can pass from one to the other. Carbon dioxide diffuses through the membrane of the mesophyll cell and enters the dark reaction by being fixed into oxaloacetate, a 4-carbon compound. Then, NADPH2 approaches the oxaloacetate and converts it to malate, also with 4 carbons. And then, to the great surprise of the researchers who first noticed it, malate diffuses not only out of the chloroplast, but right out of the mesophyll cell and into the bundle sheath cell, where it promptly enters a chloroplast. Here, malate re-energizes NADP to form NADPH2. In so doing, the malate breaks into a 3-carbon compound called pyruvate, and to the further amazement of biologists, carbon dioxide. Now, it may seem to be a pointless exercise to pick up carbon dioxide and then release it without even using it, but there is a point. The carbon dioxide released by the malate then enters the Kelvin cycle. And as we know, it is the shortage of carbon dioxide that limits the efficiency of the Kelvin cycle, and consequently the production of glucose. If we look closely at the part of the dark reaction in the mesophyll cell, we see that that whole portion of the reaction is, in reality, simply a pump that ensures an abundant supply of carbon dioxide for the Kelvin cycle. There is also a second mechanism in C4 plants that improves efficiency, and this is it. After the pyruvate diffuses into the mesophyll cell, it encounters ATP and becomes phosphoenol pyruvate, or PEP. It is PEP that picks up carbon dioxide, forming oxaloacetate. Since PEP has a low affinity for oxygen, it eagerly awaits the arrival of carbon dioxide, which certainly isn't the case with the RUDP in C3 plants. As we said earlier, C4 plants thrive in tropical and desert environments, far better than C3 plants. The explanation is now within our grasp. Special pores in the leaf surface, called stomata, regulate the movement of liquids and gases in and out of the leaf. In the hot sun, these pores very nearly close to prevent excessive loss of water to the air. But this creates a tremendous problem for the plant. Now, the diffusion of carbon dioxide into the leaf is severely restricted. C4 plants, with their carbon dioxide pumps, can easily cope with the small amounts that do manage to trickle through. But C3 plants need all the carbon dioxide they can get. So choking off the supply is intolerable. Thus, in hot regions, when carbon dioxide intake is diminished, C4 plants thrive where others fail. In temperate zones, where there is little need to close the stomata, both types of plants flourish. This is the towering redwood, Sequoia sempervirens, which grows in the thin belt along the northern Pacific coast of the United States. These redwoods are the world's tallest conifers. One specimen has been measured at nearly 120 meters, as tall as a 40-story building. At this height, and their extensive root systems allow them greater access to minerals and water. But being tall also has its disadvantages. The needles of the giant redwood, high above the forest floor, constantly lose water to the atmosphere through the process of transpiration. Transpiration occurs through special pores called stomata. Each stoma regulates the exchange of gases between the plant and the atmosphere. The diffusion of carbon dioxide in and oxygen out. And since the concentration of water vapor is usually higher inside the plant than outside, water vapor also diffuses out. This water is constantly replaced by a complex system of transports called the xylem. The xylem consists of reinforced, stiff cells, which help the tree withstand intense storms and winds. It extends from the root tips all the way up to the ends of the leaves. In the trunk, the innermost part of the xylem is heartwood and is made up of older, inactive cells. These cells have been completely filled with pigments, resins, tannins, and gums, providing added strength to the tree and protecting against insect pests. The thin outer layer of xylem or sapwood is active and consists of elongated cells with many openings or pits. Pits are particularly numerous on the tapered ends where one cell meets the next. Continuous vertical pathways are thus formed, enabling water and minerals to be transported up to the leaves. Since, in a large tree, several tons of water are being transported at any one time, a vast number of these pathways are needed. But just how a tree manages to raise water more than 100 meters above the ground is an astounding feat of engineering. Contrary to what we might think, there is no significant pumping action from osmotic pressure in the roots. Instead, plants use the unavoidable loss of water during transpiration to their advantage. As water molecules are constantly lost through the stomata, replacement molecules are drawn up through the xylem pathways. Current theories suggest that the key to this amazing accomplishment is a unique property of water. But to fully understand this, we need to look at an individual water molecule. Two hydrogen atoms are joined to a single oxygen atom at a bond angle of 104.5 degrees. Because of this asymmetry and the fact that oxygen has a stronger attraction for electrons than hydrogen, water is a polar molecule. It has a negative charge on the oxygen side and a positive charge near the hydrogens. The oxygen atom, with its slight negative charge, is attracted to hydrogen atoms in the cellulose of the cell wall. So, the molecules adhere to the sides and prevent the column of water from pulling away and breaking. Since, once broken, that particular xylem pathway would almost certainly cease to function. Even more important, the hydrogen atoms, with their slight positive charge, are attracted to the negative oxygen atoms of the other water molecules, drawing the molecules close enough to form hydrogen bonds. This effectively binds the water molecules together, so that when one is pulled up, others move along with it. And the loss of water through transpiration creates a tremendous pull on the water in the xylem columns. This binding force, called cohesion, gives a 1 millimeter wide column of water as much tensile strength as a steel wire of the same diameter. Yet as powerful as the forces of adhesion and cohesion are, they are not capable of filling an empty column 100 meters tall. Botanists have determined that in the spring, as the new layers of xylem tissue begin to grow, the cells draw water laterally from the older cells. This keeps the new cells filled as they grow to full size. At maturity, the end walls become more permeable as the pits develop, and the transport system becomes operational. The whole intricate water transport system begins with the fine single-celled root hairs, which spread extensively throughout the soil. Minerals such as nitrates, phosphates, and potassium, essential for plant growth, are dissolved in the groundwater. A number of observations indicate that roots absorb mineral ions through the process of cation exchange. The root hairs selectively absorb the mineral ions, and water follows by osmosis to reduce the concentration gradient. Once inside, the demands of transpiration and photosynthesis continually draw water and minerals up the xylem pathways to the leaves. Outside the xylem is a second transport system called the phloem, which extends throughout the plant, parallel to the xylem. This intricate branched network of cells distributes the carbohydrates produced in photosynthesis to all the active cells in the plant. In the leaves, the phloem picks up the carbohydrates by active transport. Additional water is drawn in from surrounding cells by osmosis, increasing the hydrostatic pressure within the phloem cells. At the other end of the system, carbohydrates are removed for storage and cell nourishment. Water is also removed, reducing the hydrostatic pressure within the cells. So, a continuous flow of carbohydrates from source to area of need is maintained. Water and carbohydrate molecules pass from one phloem cell to another through the specialized end walls of each cell. The end wall, containing numerous perforations or pores, is called a sieve plate. If damage occurs and the cell walls are ruptured, a special protein moves quickly to the pores and plugs them, cutting off the flow in much the same way as clotting blood seals a wound. The two transport systems, xylem and phloem, crucial role in the competitive struggle for the lion's share of sunlight, enabling plants to ascend to greater and greater heights. This is the towering redwood, Sequoia sempervirens, which grows in the thin belt along the northern Pacific coast of the United States. These redwoods are the world's tallest conifers. One specimen has been measured at nearly 120 meters, as tall as a 40-story building. At this height, and their extensive root systems allow them greater access to minerals and water. But being tall also has its disadvantages. The needles of the giant redwood, high above the forest floor, constantly lose water to the atmosphere through the process of transpiration. Transpiration occurs through special pores called stomata. Each stoma regulates the exchange of gases between the plant and the atmosphere, the diffusion of carbon dioxide in and oxygen out. And since the concentration of water vapor is usually higher inside the plant than outside, water vapor also diffuses out. This water is constantly replaced by a complex system of transports called the xylem. The xylem consists of reinforced, stiff cells, which help the tree withstand intense storms and winds. It extends from the root tips all the way up to the ends of the leaves. In the trunk, the innermost part of the xylem is heartwood and is made up of older, inactive cells. These cells have been completely filled with pigments, resins, tannins, and gums, providing added strength to the tree and protecting against insect pests. The thin outer layer of xylem or sapwood is active and consists of elongated cells with many openings or pits. Pits are particularly numerous on the tapered ends where one cell meets the next. Continuous vertical pathways are thus formed, enabling water and minerals to be transported up to the leaves. Since, in a large tree, several tons of water are being transported at any one time, a vast number of these pathways are needed. But just how a tree manages to raise water more than 100 meters above the ground is an astounding feat of engineering. Contrary to what we might think, there is no significant pumping action from osmotic pressure in the roots. Instead, plants use the unavoidable loss of water during transpiration to their advantage. As water molecules are constantly lost through the stomata, replacement molecules are drawn up through the xylem pathways. Current theories suggest that the key to this amazing accomplishment is a unique property of water. But to fully understand this, we need to look at an individual water molecule. Two hydrogen atoms are joined to a single oxygen atom at a bond angle of 104.5 degrees. Because of this asymmetry and the fact that oxygen has a stronger attraction for electrons than hydrogen, water is a polar molecule. It has a negative charge on the oxygen side and a positive charge near the hydrogens. The oxygen atom, with its slight negative charge, is attracted to hydrogen atoms in the cellulose of the cell wall. So, the molecules adhere to the sides and prevent the column of water from pulling away and breaking. Since, once broken, that particular xylem pathway would almost certainly cease to function. Even more important, the hydrogen atoms, with their slight positive charge, are attracted to the negative oxygen atoms of the other water molecules, drawing the molecules close enough to form hydrogen bonds. This effectively binds the water molecules together, so that when one is pulled up, others move along with it. And the loss of water through transpiration creates a tremendous pull on the water in the xylem columns. This binding force, called cohesion, gives a 1 millimeter wide column of water as much tensile strength as a steel wire of the same diameter. Yet as powerful as the forces of adhesion and cohesion are, they are not capable of filling an empty column 100 meters tall. Botanists have determined that, in the spring, as the new layers of xylem tissue begin to grow, the cells draw water laterally from the older cells. This keeps the new cells filled as they grow to full size. At maturity, the end walls become more permeable as the pits develop, and the transport system becomes operational. The whole intricate water transport system begins with the fine single-celled root hairs, which spread extensively throughout the soil. Minerals such as nitrates, phosphates and potassium, essential for plant growth, are dissolved in the groundwater. A number of observations indicate that roots absorb mineral ions through the process of cation exchange. The root hairs selectively absorb the mineral ions, and water follows by osmosis to reduce the concentration gradient. Once inside, the demands of transpiration and photosynthesis continually draw water and minerals up the xylem pathways to the leaves. Outside the xylem is a second transport system called the phloem, which extends throughout the plant, parallel to the xylem. This intricate branched network of cells distributes the carbohydrates produced in photosynthesis to all the active cells in the plant. In the leaves, the phloem picks up the carbohydrates by active transport. Additional water is drawn in from surrounding cells by osmosis, increasing the hydrostatic pressure within the phloem cells. At the other end of the system, carbohydrates are removed for storage and cell nourishment. Water is also removed, reducing the hydrostatic pressure within the cells. So, a continuous flow of carbohydrates from source to area of need is maintained. Water and carbohydrate molecules pass from one phloem cell to another through the specialized end walls of each cell. The end wall, containing numerous perforations or pores, is called a sieve plate. If damage occurs and the cell walls are ruptured, a special protein moves quickly to the pores and plugs them, cutting off the flow in much the same way as clotting blood seals a wound. The two transport systems, xylem and phloem, crucial role in the competitive struggle for the lion's share of sunlight, enabling plants to ascend to greater and greater heights.